In this video, we are going to do an introduction to biomolecules. I'm going to be discussing selected topics of chapter 24, 25, and 26. First, I want to establish that the study of biomolecules is very intertwined with biochemistry. Biochemistry is the science that studies life at the molecular level. When it comes to biomolecules, understand that there are four main biomolecules that are understood in biochemistry. Those include carbohydrates, lipids, amino acids, which are the subunits of proteins, and nucleic acids. Out of these four biomolecules, we are only going to be discussing four in the following content of these videos. First, we are going to start by talking about carbohydrates. Carbohydrates are also known as sugars, and they are polyhydroxyaldehydes or ketones. Carbohydrates are also known as saccharides. And understand that the term carbohydrates comes from the idea that these biomolecules are going to have the general formula CnH2ON for the monosaccharides. There are some larger carbohydrates that also have this general formula. N refers to an integer and it's going to be anywhere from 3 to 8 for the monosaccharides. And monosaccharides simply are going to be a single unit of carbohydrate, as we're going to be discussing. Now, understand that monosaccharides are synthesized from the combination of CO2 and water. And there are some organisms like plants, algae, and cyanobacteria that have this process happen. And they utilize energy that comes from sunlight to produce, for example, glucose, which is the most common carbohydrate monosaccharide because it's a single unit out there. You can see that by definition, as I just mentioned, the polyhydroxy comes from the idea of the several hydroxy groups that are present in carbohydrates. And glucose specifically has an aldehyde motif. Now, understand that when it comes to us as humans, we cannot utilize CO2, water, and sunlight as an energy resource to synthesize glucose. Now, we have the ability of synthesizing glucose, but it's going to be more specialized. We do it from non-carbohydrate sources, such as amino acids and the glycerol portion of fats, which these are things that we are going to be talking about later in the chapter. You do not have to memorize all these processes of how these compounds result in the formation of glucose. I just want to illustrate the complexity of how, as humans, we actually undergo many reactions to synthesize glucose. Understand that monosaccharides, which is one unit of carbohydrate, which in other words are simple sugars, have multiple chiral centers. Now, understand that whenever we are writing sugars, specifically monosaccharides, in open form, we're going to use Fisher projections. And you can see illustrations of Fisher projections in the two structures that we have in the bottom. Now, a Fisher projection, in essence, what is illustrating is that at every single intercept, even though they're not written, it is implied that there's a carbon atom. So as you can see in the molecule of glucose, we have carbon atoms at the body of that Fisher projection. In fructose, as you can see, we also have carbon atoms. Now, The chiral centers that are present in the molecule, I'm just going to highlight them in blue, are going to be those carbon atoms that I actually wrote in. Remember that when it comes to the chiral centers, those chiral centers are going to have 4 different groups. Okay? Now, the classification of monosaccharides also is extended from that. The basic idea of classifying monosaccharides includes what is the carbonyl functionality that is present in your molecule and how many carbons are in there. So, depending on the carbonyl functionality, which is the first 2 bullet points, you can classify sugars, monosaccharides, as aldose if it has an aldehyde and as a ketose if it has a ketone group. In terms of the number of carbons, we are going to use very specific prefixes to denote the number of carbon atoms. So if you have 2 is di, 3 is tri, 4 is tet, and then from 5 on, we are going to classify it based on Greek prefixes. One thing that I want to establish is that for monosaccharides, the majority of monosaccharides have 3 to 8 carbons in length. So the prefix di is actually not typically utilized. Now, if we have a monosaccharide, understand that they're going to have specifically a suffix that is OSE. So that's how you know that you're talking about a carbohydrate, because the name is going to end in OSE. So if we're trying to build the classification of a particular monosaccharide, the first thing that we're going to do is determine if it's an aldehyde or a ketone. So for an aldose, you're going to use ALDO. For a ketose, we're going to use KETO. So if we look at the examples that we have in the bottom, the first carbohydrate, or the first monosaccharide, I should say, is going to be, because of the aldehyde present in the functionality, it is called an aldo. Because of the 5 carbons, it's called pent, and then the ending is going to be OSE because we're talking about a monosaccharide. If we look at the next example, then there is a ketone functionality. And then this ketone functionality designates that this is going to be a keto because it has 6 carbon atoms in total, is a hex, and then because it's a monosaccharide or a carbohydrate in general, is going to end in OSE. So this is a keto hexose. The other classification that we have for monosaccharide, it has to do with chirality. As we already established, there are chiral centers present in these molecules. So for example, here we have the example of the simplest aldose, which is known as glyceraldehyde. In the case of glyceraldehyde, there is one chiral center, which I'm highlighted in green. And when we look at this chiral center, in essence, we can see that one designation is D-glyceraldehyde, the other designation is L-glyceraldehyde. Now, understand that this D and L notation, it is actually related to RNS configuration. Because, as you can see here, a D sugar means that the configuration of the furthest chiral center, okay, is R. While an L sugar has an S configuration at the furthest chiral center. Now, what chiral center are we talking about? As you can see here, and I'm highlighting in blue, the chiral center that is going to determine D or L notation is going to be the chiral center that is the farthest away from the carbonyl, carbon in the molecule. So for example, if we see the example that we have for glyceraldehyde, notice that the D configuration is because the OH is to the right. And that's the quicker way to do it. As long as you have the aldehyde functionality on the top, you have the CH2OH in the bottom, and you have in the middle of your Fisher projection the H in the OH. If the OH is to the right, then that is D-glyceraldehyde. If your OH is to the left, then that is L-glyceraldehyde. So if we look at the different carbohydrates given in the bottom of the slide, D-ribose, D-glucose, and D-fructose, you can see that the OH at the chiral carbon that is the farthest away from the carbonyl functionality is towards the right. Notice that most sugars in nature are D configuration. And that's just one observation from experimental data. Now, one of the things that it is observed is that these monosaccharides are not found in open form, which is the drawing that we observe in the Fisher projection. One of the things is that we learned previously in the aldehyde and the ketone chapter that if you have an aldehyde or a ketone in the presence of an alcohol, one of the things that can form is a hemiacetal. Now, for the most part, hemiacetal formation is not favored, but in the case of a carbohydrate, understand that this is going to be an intramolecular reaction. So this is going to result in the formation of a cyclic hemiacetal. And in the case of monosaccharides and other carbohydrates, that is favored. Specifically, we are going to see the formation of 5 or 6 membered rings. So understand that monosaccharides undergo ring closing, hemiacetal formation. And if we observe overall what is happening to the molecule, and I'm just going to highlight the chiral centers, we have in the beginning, in the Fisher projection open form, 1, 2, 3, 4 chiral centers. So in the second drawing, what we can see, that corresponds to still the open form, but we can directly see which ones are the atoms that are going to be participating in the ring closing of the system. So specifically, the OH that it is involved in determining the D or L configuration for your carbohydrate is going to be cyclizing by forming a new single bond with the carbon in the carbonyl functionality. If you have, for example, in this case, we have an aldohexose undergoing cyclization, what we can observe is that if we count now at the chiral centers that are present in the molecule, we started with 1, 2, 3, 4, and the new sigma bond, in other words, the new bond that is formed right here between this oxygen atom and the carbon in the carbonyl creates a new chiral center. And that chiral center is known as the anomeric carbon. Understand that the equilibrium of this system is going to greatly favor that cyclic form. And when it comes to aldohexose, we are going to be forming a 6-member hemiacetal ring of that saccharide, and that specifically is going to be called a pyranose ring. So if we look closely at what the reaction is doing, and we're trying to figure out how do we go from the Fischer projection to the closed form that is called a Hayworth projection. It is actually fairly easy to understand how we have that inner conversion. Okay. Now, these are the steps in order, if you are given a Fischer projection for an aldohexose, how to draw the Hayworth projection. Step number 1. You are going to take your Fischer projection and you're going to lean it towards the right 90 degrees. So what that means is that every single atom that it is on the right of the molecule is going to point down. Any atom that it is to the left of the molecule is going to point up. That does not include the carbonyl or the CH2OH. So let's just do that drawing first. So if I take my monosaccharide, and here is where I have my aldehyde, and here is where I have my CH2OH. In the middle, I'm going to have 1, 2, 3, 4. Okay? And I'm just going to keep the numbering the same. Where the carbonyl carbon is number 1, then in the middle I have carbon 2, carbon 3, carbon 4, carbon 5, and carbon 6. Then, as I lean it on carbon 2, my OH is pointing down, my H is pointing up. On carbon 3, H is pointing down, OH is pointing up. On carbon 4, OH is pointing down, H is pointing up. And lastly, on carbon 5, OH is pointing down, H is pointing up. In the next step of creating that Hayworth projection, I know that overall, this OH is what bonds the carbon in the carbonyl. Okay? So if I'm trying to draw the cyclization, remember that overall, as we learned previously, I'm just going to erase the dotted lines. Let me write the bonds. One of the lumpers from the oxygen is going to go and attack that carbon atom. Okay? Now, understand that this specifically is going to overall happen in under acidic conditions. So if we are under acidic conditions, we're assuming that this oxygen is protonated in a previous step. Again, all of this, when it happens in nature, is all happening in the same vessel. So if I have a protonated aldehyde and then I am attacking from this oxygen onto that carbon, then I'm going to put the electrons up to the oxygen. And overall, understand that what is happening is that I am wrapping, okay, these atoms around in order to cyclize. So the attack of this molecule can happen through the front, okay? So you can imagine on top of the paper or behind the paper, okay? So what does that mean overall? That in the second step, what I'm going to do is write that folded form that we see that is still open, where this is my OH, this is my aldehyde. So if I number the atoms, this is 1, 2, 3, 4, 5, okay? And on carbon 5, here is the CH2OH. So technically this is carbon 6. Then on carbon 2, and I'm just going to color code it to make it easier. If we look at carbon 2, the OH is pointing down. On carbon 3, the OH is pointing up. On carbon 4, the OH is pointing down. Carbon 5 is what's going to be involved in the cyclization. So again, we can envision that this is protonated, that this is going to attack at the carbon atom. The electrons are going to go up to the oxygen. And that's what results in the Hayworth projection. I am not including the hydrogens. Carbon 2, OH is down. Carbon 3, OH is up. Carbon 4, OH is down. On carbon 5, the CH2OH is pointing up. And remember, the OH that is attached to carbon 5 is what makes that bond. Now, understand that at this point, I'm going to draw the new chiral center or the anomeric position as a squiggly line. And the reason is because this new chiral center that is created here, the anomeric position, can have different conformations. So as I just mentioned, when it comes to the stereoisomers that come from the cyclization of aldohexose, because the OH that binds to the carbonyl functionality in the aldohexose can attack from the top of the molecule or from behind the molecule, then we're going to have two different configurations for the Hayworth projection, okay? Now, one is going to be known as the alpha anomer. And as you can see, the anomeric OH group is trans to the CH2OH group. And that's the molecule that we have on the left. We also have the beta anomer. And the beta anomer is going to be the anomeric OH that is cis to a CH2OH. Now, when it comes to this, I actually designed a little acronym that always guides me to know which one is the alpha and which one is the beta, okay? And what I utilize is the little acronym Buddha, which means beta, up, down alpha. What does that mean? That if I have a Hayworth projection and I look specifically at the anomeric position, which I'm highlighted in green right now. Let me use a different color. Let me just use orange. So if I look at the anomeric position, okay, I know that my beta configuration is going to have the OH at the anomeric position up. As you can see, the OH is up at the anomeric position. While if I have an alpha configuration at the anomeric position, my OH is going to be pointing down. And that is another simple way to distinguish between the beta anomer and the alpha anomer. So understand that when it comes to the Hayworth projection or the chair conformation for these structures, understand that it can be drawn as a flat molecule, which is what I just illustrated, or as a 3D conformation. Understand that the oxygen ring always is drawn on the upper right rear position. Okay. Understand that D-glucose is going to be the only sugar where all the groups are actually equatorial to one another, and that is the most stable sugar. Okay, and it's actually the most abundant in nature. So as you can see, that's the one that is illustrated in the following slide.